Alright, we're going to do a quick lesson on viruses now. Viruses are non-living entities that are active inside of living things. And the non-living part is kind of weird for people to understand because viruses look an awful lot like they might be living things. They get into cells, they can run, a program for replication and so on. And you kind of need to think of a virus like a robot. Viruses, like robots, can run programs, but they don't really carry out all the life functions that an actual living thing carries out, and therefore a virus can't be considered living, the same way that a robot can't be considered living. So the first thing I wanted you to notice here as you look at this is these are three different types of viruses, and I just chose three different pictures. This is the flu virus here. This is a T4 phage. This is a virus that gets inside of a bacterium. This is tobacco mosaic virus, so viruses can even attack plants. The point here, what I'm trying to show you is there's a lot of variety in viruses. Okay, no two viruses are necessarily put together the exact same way. And this is because the virus is specific to its host. The structures that you see here, especially the protein coat, like you see here, the capsid, the outside of this particular virus, it's adapted to get into a particular host cell. And each virus can only get into a certain host cell because its protein coat matches some receptor on the host cell's surface. And so there's a lot of specificity in viruses. Okay, so viruses are specific to their host cells, and therefore, because they're so specific, there's a lot of varieties of viruses out there and a lot of different shapes and things like that that you'll notice with a virus. What you need to know is, how does a virus reproduce? And there's a simple cycle, and then there's variations on that cycle. And the simple cycle is here. That's the Lytic cycle, which is what we're seeing here. So we'll start here with the Lytic cycle. This is the attachment phase of the Lytic cycle. So here's a little T4 virus attaching to a bacterium. It attaches and it injects its DNA into the host cell. Okay, so that's kind of step one. The virus latches onto a cell and injects its DNA. Sometimes the virus will go in on its own. Sometimes it just sends its DNA in. That DNA gets integrated into the host cell's chromosome and then starts to run a program. Okay. Now, the reason why the virus can get in, again, is because the virus matches a particular protein on the host cell's surface. So it's kind of like the virus is in disguise, and it tricks the host cell into letting it in. So the host cell thinks that this is some harmless chemical from someplace else and lets it in. Once you have a Lytic cycle virus, once that virus gets in, that host cell will never be the same. As soon as the virus is in, it starts replicating viral DNA. It starts transcribing and translating it so that you end up with little new protein coats being assembled, and that's what you see here. So the red are the new viral pieces of DNA, the blue are the new protein coats. Then those get assembled into new viruses until the cell is so packed full of viruses that it literally bursts open, and that's called lysis. And that's why this is the Lytic cycle of viruses. And then once that happens, these viruses go out and attach to other cells, and that's why it's called a cycle. Okay. So this cycle, the basic cycle, the Lytic cycle, is when the virus attacks a cell and is immediately active. And viruses that do this are things like the flu, influenza, like you saw in your video. Also things like rhinovirus, which causes the common cold, most of the viruses that you get sick with during the year. Now, there is a variation on that, and that's the lysogenic cycle. And the lysogenic cycle starts the same way. But here what happens is there's a complicated step. When the virus attacks and then injects its DNA into the host cell, that DNA can actually hide in the lysogenic cycle inside of the host cell for a very long period of time. So instead of becoming active right away, a lysogenic virus actually just kind of stays inside of the host chromosome and hides there. And then when the host does mitosis and replicates its DNA, it replicates the viral DNA as well. So that little red piece of DNA, that viral DNA, gets replicated. So now instead of one cell with virus, you've got two cells with virus. And depending on how long that cell stays inactive, the virus stays inactive, it'll continue to replicate and replicate. So then you get 4 cells, then 8 cells, then 16, then 34 cells, and pretty soon you've got this patch of thousands of cells that are all infected with a virus, all from one viral infection right back here. So the virus just kind of hangs out, keeps replicating as the host does mitosis. Then what happens is the virus is triggered, okay, by something. There's a trigger, and the triggering event causes some of the cells containing virus to go active. So the virus inside of some of the cells that are infected starts to go active and then finishes up the steps that you see in the Lytic cycle. So it starts here, kind of takes a detour down here, and then goes back through the Lytic cycle. Now here's the thing about these lysogenic viruses. Because not all of the cells that contain virus become triggered, you never get rid of one of these viruses. There's always some cell somewhere in the body that's got some virus in it left over in this lysogenic cycle. What that means then is once you have these viruses, you have them forever. You're never going to get rid of them. So things like herpes, which there are actually a bunch of herpes. Chickenpox is a variety of herpes, but then you also have the herpes that causes cold sores, the herpes that causes sores on your genitals, things like that. This is a virus that once you get it, it's lysogenic. So once it's in your system, it stays in your system forever and ever and ever and ever. You can never ever get rid of one of these viruses because the virus is always hiding, okay? Your immune system can't detect viral DNA once it's integrated into your own DNA inside of your cells. So once you have that, you've got that forever. Then there's sort of one last variation on the virus theme, and let's go to the next slide for that. All right, so the last variation on the viral cycle is that of the retrovirus. So we're dealing with retroviruses here. And an example of a retrovirus is HIV. Now, retroviruses tend to be lysogenic, okay? But retroviruses have a problem, and the problem is that instead of DNA, a retrovirus contains RNA. All right, so viruses pretty much at their simplest are just pieces of genetic material covered in protein. Sometimes they have an envelope that surrounds them, sometimes they don't, but they're usually relatively simple. A virus like HIV, because it has, instead of DNA as its genetic material, RNA, and because it's lysogenic, it wants to insert that genetic material into your chromosomes, but your chromosomes are made of DNA, okay? So what the retroviruses do is, if you come down here, you see here's HIV attaching to the cell, going into the cell, and here is the viral genetic material right here, okay? That's RNA. It injects, along with the viral genetic material, it injects these little reverse transcriptase enzymes here, okay? So right there on the genetic material, you can see a reverse transcriptase, and because it ends in ase, it's an enzyme, right? And it's going to do reversive transcription. Now remember, transcription is making RNA from DNA, right? So if we're going to reverse transcription, what's happening is you're taking an RNA template and you are using it to make DNA, okay? So in reverse transcription, that's what's happening. So the virus uses this little enzyme, uses its RNA to construct DNA molecules, And the DNA transcript then, whoops, sorry, the DNA transcript is in red here. This DNA transcript then would be sent to integrate itself into the chromosomes of your T cells. Okay, so that's how a retrovirus works. So instead of just getting into your cells and hiding there, before it can actually get in and hide, it's got to take its own genetic material and use it to make DNA that can be integrated into your chromosomes. That's pretty much it for viruses. You know, there's not a whole lot that you have to know. You do need to understand, though, that they are active inside of living things. You need to kind of understand how they get into your cells and why they cause trouble. Go back and rewatch that flu video if you need to. That's a really good example showing you how viruses impact the body.